to our YouTube channel. Today we'll be learning something else that's very vital, and that's cell membrane transport. Cell membrane transport. So we'll dive right into it. Now, the membrane, we learned about the structure of the cell membrane, and we established that some things can pass, things that are lipid soluble, other things find it hard passing just because they are water soluble. Now, how does the body navigate transporting things that are lipid soluble and then also transporting things that are water soluble? So now that's what we're learning today, and that's what we call cell membrane transport. Now, transport across the cell membrane is by two primary ways. One is diffusion and the other one is called active transport. In today's class, we'll be learning about diffusion. Now, what is diffusion? In simple words, we always think of diffusion as anytime you move from higher to lower, from place of higher concentration to a place of lower concentration. So from what we said, diffusion is from higher to lower, so it's downhill. Now think of it this way. Now whenever you're moving from higher to lower, you are favoring the concentration difference or gradient. So, and also because I'm moving from higher to lower, I don't need energy, additional energy. Now let's use an example. Bikes, you see cars, you see bikes. If you're going up a hill, there's troubles. They might have to change gears, they might have to do different things. But whenever they are coming down a hill, you realize they do it effortlessly. You just go down. For those times, you could see bikes put off their bike, or even a vehicle can put off the ignition and then allow the vehicle to just go. It will flow on its own because it is what? Downhill. So that's diffusion. Primary things to note about diffusion, which is our topic or our focus for today, is that it is favoring concentration gradient, which of course is the difference in concentration. It is a downhill movement, and then it doesn't require additional energy. Why am I using the word additional? Because ideally moving from higher to lower, we can believe that kinetic energy is involved, but it does not require any other form of energy. Now, diffusion is of two types here when we think of cell membrane transport. Generally, it is either simple diffusion or facilitated diffusion. Now, simple in this case is that it has no need for a carrier protein. So either the stuff just passes directly through the membrane, or it uses a channel which is still like a free passage, but then it does not require a carrier protein. You remember who we said saved this role of carrier proteins? Integral proteins. Yeah, I know you remember that. Okay, so if you've not heard that before, you can go back to our former video. It was clearly stated there. Now, so that's that. So we either have simple or facilitated. So facilitated, like we said, requires carrier protein. But what is common to these two transports? It is always from higher to lower. So let's discuss simple diffusion. Now, simple diffusion. Simple diffusion, like we said, is either of, okay, simple diffusion, like we said, is two. It could either be directly through the spaces or through the membrane, directly through the lipid layer of the membrane. And of course, people that can do this are going to be lipid soluble substances. People that can actually pass through the lipid layer very freely, directly, are lipid soluble substances. But then on the other hand, I could also have them. We could also have transport through channels. Of course, we call them watery channels, we call them porins. So we could also have things flow through channels, watery channels called aquaporins. Okay, now, so we discuss each of them. Of course, this one from the name, that'll be water, will be what will be passing through. And then also, the other thing that can pass through here will be water-soluble molecules. So let's pick the first one. When we say things are transported directly through the lipid layer of the cell membrane, those things should be lipid soluble. A key example, so I could take star one. A key example of things that are lipid soluble, we could use CO2, oxygen. Those are our respiratory gases. We could even have nitrogen being lipid soluble. It's also lipid soluble. And then alcohol. So this is why someone that is someone gets intoxicated with alcohol, you could have the effects quickly. Now, we need oxygen transport to be free. Oxygen is lipid soluble because the cells readily need oxygen. So I don't even need him to be struggling to enter the cell. And it is believed that the rate at which oxygen enters the cell, it enters so fast that it's as if the membrane never existed. You know, when you are so free that you can just enter a place so freely, that's how quickly oxygen can enter. So these are the substances that can actually cross very fast. And please understand, also take note that the ability to, or the speed, or the rate of their diffusion through the lipid layer depends on their lipid solubility. So how soluble are they in lipids? How soluble are they in lipids? That will also greatly affect the rates at which they diffuse. So that's about simple diffusion through the lipid membrane. So please, take note that there are lipid soluble substances of which I've given you examples, and then these guys, their rate is dependent on their solubility in lipids, which I said oxygen is primary. Now we go to people that go through channels. Now the first thing that you take note of here is that these guys, remember who channels are. Integral proteins that actually cross the entire length of the membrane. We call them transmembrane proteins. Now these channels here allow primarily water to pass through it. But aside from water, we could also have water-soluble molecules. So these molecules could either be, we could also even have water-soluble ions, like potassium, sodium, all those are water-soluble ions, so they can also pass through the membrane. But aside from that, water itself primarily can pass. But the thing we should note about this second guy, so here remember the rate was based on lipid solubility. But this particular second guy has something we call selective permeability. So you know when I have a channel, but the channel is selecting people that can pass. This selectivity is actually dependent on things like diameter of the channel. Now if the channel is narrow, that means only things that fit into this size. So some substances can be bigger than it and they cannot pass. Think of a doorway, and then you have a particular guy that's, okay, let me say someone that is healthy, let me not use the word fat, but then you realize the person cannot pass through it. Or you think of a particular this can pass through because it fitted into the hole, the diameter. But think of it was this narrow, I wouldn't have this pass through. So that's one of the ways we convey selectivity. So I'm saying that these guys convey they convey a particular thing we call selectivity. And how do they convey it? One, by diameter of the pores. Then apart from the diameter of the pore, it could even be the shape of the pore. Of course, if the pore is circular, it will allow circular things to pass. If it is rectangular, other things like that could pass. And okay, so we realize we have the diameter of the pore. Of course, diameter will affect the size of what is entering. And then we could also even have the shape. And likewise, we could even have the electrical charges within the channel. Now, most times we realize that channels have negative charges. That means for positive things to come in, if negative things wants to come in, to repel those things, but it will allow positivity to come in. I believe you're understanding me. So when they are aligned by negative, negative on like poles repel, on like attract. So negative will repel any other negative, but that negative will accept positive to come in. And here is that you now realize that, for example, some channels, potassium more, the other one more. So that's that. Another thing that's important, another way that ion permeability is regulated, or controlled is by way of things we call gates. Now think of what is a gate. What is a gate? Something that limits you from entering a particular house. A compound we say is fenced and has gates. It limits you from entering that place. So ion permeability can also be controlled by gates. So hence we call these kind of channels, the kind of channels that can be controlled by gates, we call them ion gated channels. Now there are two primary types of channels that are gated, or two primary types of gates. One is called voltage gated and the other is called ligand gated. Voltage gated, ligand gated. So when I look at a channel that is voltage gated, that means it allows only, so please take note, voltage gated. So this channel and ligand gated. Voltage gated only opens when electrical charges. So there's something that will cause a gate to open. Now you have a bouncer at the gate, and it's either you are with your tickets or you have connection. So if you are not with tickets, you are not entering. So that's the king here. So voltage gated channel, the voltage, what opens the gate is electrical charges. So okay, charges are generated, that's what opens the gate. But on the other hand, ligand-gated channel, what opens the gate is a chemical. An example, we'll talk about this in details as we go on in other lectures, but the main thing voltage-gated channel helps in is in action potential. So those channels, when voltage-gated channels open, most times in action potential will happen because the voltage generator will keep opening more channels and channels and channels. Whereas ligand-gated is, for example, is seen in the case of acetylcholine, when acetylcholine binds either to a nerve terminal ending or to a muscle. So it's really important in nerve transmission of impulses from nerve to another nerve, and then transmission of impulses from nerve to a muscle, of course, in excitation contraction. That is so that the muscle could contract. So that is that about simple diffusion. So we now go into the next part, which is going to be facilitated diffusion. So now, facilitated diffusion, please remember I said diffusion can either be simple or facilitated. These guys, the simple did not require any form of carrier protein. So what they used was either directly through the membrane or they used channels. Remember I told you carrier proteins and channels are different. Carrier proteins, channels, but both of them are transporters. So, but these guys require carrier proteins. And because they require carrier proteins, there's something you must take note of here. Because they require carrier proteins, they are now selective, very selective. You know, the selectivity here was in terms of size. This one I cannot even call it really selective because when you look at the size, okay, if bigger size, I might not really know is that potassium, is that calcium. I just need to know that, okay, you are small, you are large. But for this other case where I talked about selectivity, they can only bind to a particular type of thing. If it is not that stuff, they cannot bind. So that's facilitated diffusion. Yes, it is still diffusion higher to lower, but then they require carrier proteins and then they are very selective. For that reason, they are called carrier mediated diffusion. So they require carrier proteins and then they are very selective. They only bind to specific things. And then, of course, what also happens is that after I bind, remember these things are not transmembrane. Once the particular stuff to be transported binds, gradually the body changes the conformation. So the membrane will change its conformation and then internalize. So it's more like you bind, I change my conformation and then push you to the other side. So think of a ferry, you pay the boat driver, he picks you inside the vehicle, takes you to the other side and then drops you. So that is that about facilitated. So what have we learned in our lecture today? Diffusion. That there are two primary ways of transporting anything across the cell membrane. Primarily diffusion and also active transport. Now, diffusion is what we took to learn today, which is actually movement from higher to lower. Higher to lower. That's why we call it downhill. That's why we say it's favoring concentration gradients. I'm always moving from higher to lower. That's the normal way of things, from where there is more to where there is less. And then it does not require additional energy. We now want to talk about the fact that diffusion can either be simple or it can be facilitated. Now, what is simple diffusion? Simple is when I don't require a carrier protein. So generally here, you can either go through the membrane very freely like lipid-soluble substance. This is my zone. Or I could use channels like water. Water is important. And then other things can also use channels. Other things can also use channels. I mentioned ions. Now, when we come to facilitated diffusion requires carrier proteins that are very selective, and then we also call it carrier-mediated diffusion. I mentioned that why they are selective is that they can only bind to a particular type of thing. Please, an example here are our glucose transporters. So when we transport glucose, and even when we transport most amino acids, so glucose transporters, we hear of GLUT4, for example, that is dependent on insulin. We see it in skeletal muscles and adipose tissue. So they have their role, they help in transport. If it is not glucose, that transporter cannot carry in. Please don't mistake it. I said transporters are not carrier proteins alone. Transporter could either be anything that can carry something from one side to another. So it could be a channel, it could be a carrier protein. But when the transporter here is a carrier protein and it's diffusion facilitated, when the transporter is a channel and it's diffusion, then we are looking at simple diffusion. So that brings us to the end of the first session of this class, which discusses diffusion. Thank you very much. I'll see you in the next class where we continue on active transport.